Hello, I'm Ron Vale, and I'd like to welcome everyone to Life Science Across the Globe, a Sister Institute seminar series. Information on these talks can be found on this slide and also on the website lifescienceacrosstheglobe.org. So thus far, we've had talks from North America, South America, and today we travel across the Atlantic Ocean to Great Britain and welcome our sister institute, the Laboratory of Molecular Biology in Cambridge. Today you're going to hear two talks. One is a science talk and one is a science culture talk where you'll also have a chance to learn about the very influential and interesting intellectual environment of the LMB. And after these talks, you'll be able to ask questions and they'll be moderated by the series organizer, Dr. Janine Stevens. So without any further ado, I'd like to welcome our sister institute, the LMB, and its director, Jan Löwe. Welcome, Jan.
Thank you, Ron. I just wanted to extend my warm welcome to everyone who's joining from all over the world. Since this is LMB's first talk in this exciting series, I just wanted to say a few words. We are, of course, delighted to be part of this endeavor. And we're also a little bit lucky, I think, because as we just discussed, LMB is running at almost 100% again, but COVID has stopped a lot of the communication that we used to have with the rest of the world. And I think it's also fair to say that international collaboration is not very popular with some people at the moment. And in that sense, something like this seminar series could really make a difference in showing how the value of cooperation and the exchange of ideas is important to scientists and how that needs to continue. I think what this series also might do is to move science communication into the 21st century, essentially with much less travel. And that's really all I wanted to say. I want to hand over to Anne Bartolotti, who is who chairs LMB Seminar Committee. And in that role, one of her main jobs is to select speakers for our named lectures, named after some of the founding fathers of LMB, Max Perutz, Francis Crick, Aaron Klug, and Cesar Milstein. She also works on a topic that's related to neurodegeneration, so she's much more competent in introducing the speakers than I am. Anne, I'm handing over to you.
Thank you very much, Jan. So when preparing for this with the LMB Seminar Committee, we came up with a very long list of possible speakers, so it was a very difficult choice. But we've asked today Michel Goedert and Sjors Scheres to present the first LMB's talk in this very exciting series because their work together exemplifies very well the type of work that LMB enables. So LMB provides a really unique opportunity to focus on important and challenging problems that often take years to solve and also fosters collaboration. And you'll hear a very exciting example of that today in Michel and Sjors' talk. So a bit of introduction on our speakers today. Michel was born and raised in one of Europe's smallest countries, Luxembourg. And he went to Switzerland to study medicine and then moved to Cambridge for his PhD. He joined the LMB in 1984 for a postdoc and was rapidly promoted group leader. He became joint head of the Neurobiology division in 2003, a position he held until 2016. His work on the molecular basis of Alzheimer's disease has received numerous awards, and I will only cite two. He was one of the co-recipients of the Brain Prize from the Lundbeck Foundation in 2018, and he won the Royal Medal of the Royal Society here in London in 2019. Sjors comes from the Netherlands, where he studied chemistry in Utrecht University, where he also gained his PhD in protein crystallography. He was then a postdoc in Madrid, in Spain, before joining LMB as a group leader in 2010. He became joint head of the Structural Studies Division in 2018. Sjors is famous for the development of the computer program Lion for high-resolution Cryo-EM structure determination, which is now very widely used. I know I will embarrass him in saying this, but because of his work, Nature featured Sjors as one of the 10 people who mattered in 2014, but I'd like to reassure him he still matters to us now. So Michel and Sjors joined forces and talent to shed light on the molecular basis of neurodegeneration, which is a problem that is, of course, of outstanding importance and that has been studied here at LMB for a very long time, with important contributions for a number of alumni, as you'll hear in Michel's talk. So today, both Michel and Sjors will present how, in the recent years, they've determined the structure of pathological amyloid filaments from brains of deceased patients. And we will start with Michel, and Sjors will follow. Enjoy.
Thank you, Anne. And thank you, Janine and Matt, for all your help. This slide shows a list of human neurodegenerative diseases. They account for the vast majority of cases of disease, and these diseases share the presence of abundant pathological amyloid assemblies in nerve cells, and for some of them also in glial cells. Alzheimer's disease is defined by two types of assemblies, the so-called plaques and tangles, the other diseases by only one, and assemblies made of the same proteins are found in multiple diseases. Tau assemblies, shown here in blue, define 7 of the 13 diseases, whereas alpha-synuclein assemblies, shown in red, define 3 diseases. These diseases are highlighted here because this talk is about the Cryo-EM structures of tau filaments and alpha-synuclein filaments from human brain. There are no approved mechanism-based therapies for any of these diseases. Alzheimer's disease and Parkinson's disease are the most common neurodegenerative diseases. Alzheimer's disease is defined by abundant beta amyloid plaques and tau tangles, whereas Parkinson's disease is defined by abundant alpha-synuclein Lewy bodies. Most disease cases are sporadic, but some are inherited in a dominant manner. Exonic and intronic mutations in the tau gene cause frontotemporal dementias with abundant tau assemblies. Missense and gene dosage mutations in the alpha-synuclein gene give rise to Parkinson's disease and dementia with Lewy bodies with abundant alpha-synuclein assemblies. And importantly, close links exist between protein assembly and the causes of inherited forms of disease. By extrapolation, formation of amyloid filaments is probably central to neurodegeneration in all cases of tau proteinopathy and synuclein proteinopathy. And filamentous assemblies form in defined brain regions from where they appear to spread through prion-like mechanisms, resulting in neurodegeneration and symptoms of disease. In 1983, Aaron Klug initiated work on Alzheimer's disease at the LMB. The goals were to find out what paired helical filaments are made of and to determine their structures. Paired helical filaments had been identified in Alzheimer's disease brains by Michael Kidd in 1963. They are the major components of tangles. In 1988, we showed that the microtubule-associated protein tau is an integral component of paired helical filaments. In 1991, Tony Crowther at the LMB showed that paired helical filaments and the related straight filaments of Alzheimer's disease are each made of 2 identical C-shaped protofilaments that are linked in different ways. And these protofilaments form the filament core, which consists of the C-terminal repeat region of tau, with the N-terminal region being unstructured and forming the so-called fuzzy coat. In 2017, we used Cryo-EM to determine the high-resolution structures of tau filament cores from Alzheimer's disease brain. In 1989, we showed that in normal adult human brain, there are 6 tau isoforms that are produced from a single gene by alternative messenger RNA splicing. Depending on the inclusion of exon 10, shown here in yellow, 3 isoforms have 4 repeats and 3 isoforms have 3 repeats. And each repeat is 31 or 32 amino acids in length. The repeats and some adjoining sequences form the cores of tau filaments. The repeats also form part of the microtubule binding domain of tau. Depending on which tau isoforms are present in disease filaments, one can divide these diseases into 3 groups. Tau filaments from the brains of individuals with Alzheimer's disease, chronic traumatic encephalopathy, and tangle-only dementia consist of all 6 tau isoforms. In Pick's disease, only 3 repeat tau is found in the abnormal filaments, whereas in corticobasal degeneration, progressive supranuclear palsy, and argiophilic grain disease, only 4 repeat tau is found in the abnormal filaments. And we have determined the structures of tau filaments so far from Alzheimer's disease, chronic traumatic encephalopathy, Pick's disease, and corticobasal degeneration. Unlike tau, alpha-synuclein exists as one major isoform. In 1997 and 1998, we extracted alpha-synuclein filaments from the brains of individuals with Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy. And we recently reported the cryo-EM structures of alpha-synuclein filaments from multiple system atrophy. And I now hand over to Sjors for the rest of this talk.
Thank you, Michel. After the cryo-EM resolution revolution, and after Xiao Dahi, a student in my lab, implemented helical symmetry in RelyOn, we thought the time was ripe to apply cryo-EM structure determination to tau filaments extracted from AD brains. This is when Michel and I started to collaborate. Ben Falcon, a postdoc in Michel's lab, extracted tau filament from the cortex regions of a brain of a 74-year-old female who had died with advanced stages of Alzheimer's disease. Negative stain imaging of these PREBs, shown on the right here, showed the same PHFs and SFs, paired helical filaments and straight filaments that Tony Crowther had observed earlier. Anthony Fitzpatrick, a postdoc in my lab, then performed cryo-EM imaging on these PREBs. He managed to solve 3D reconstructions up to a sufficient resolution to allow de novo atomic modeling. On the left here, you see false colored raw cryo-EM images of PHFs and SFs with zoomed in 2D class average regions in which one can distinguish the separation of individual beta strands which form the large beta sheets which run along the helical axis of the filaments. On the right-hand side, you can see the 3D reconstructions. Paired helical filaments have the typical variation of wide and narrow in their projection views because in the XY plane, perpendicular to the Z axis of the helical axis, the structure is much wider than it is high. Straight filaments appear straight in the projection views because their dimensions in the X and Y direction are more or less similar. The paired helical filament is made up of 2 C-shaped protofilament structures which were imaged at much lower resolution by Tony Crowther before. Because of the resolution being high enough to allow de novo atomic modeling, we could define that the ordered core of the filaments runs from valine 306 to phenylalanine 378. All the other residues of tau are in the so-called fuzzy code and are disordered. At each level in the filamentous structure, 2 identical C-shaped protofilaments, each made up of 1 molecule of tau, are related by helical symmetry with each other. This is not the case in the straight filament structure, where two very similar C-shaped protofilaments made up of the same residues pack against each other in an asymmetric manner. In this case, an additional density, which is coordinated by 4 central lysines, appears to be involved in holding the two C-shaped protofilaments together. When Anthony left for Columbia University to set up his own lab, Ben Falcon continued to image more cases of AD brains by Cryo-EM, and he was helped in this endeavor by Wenjun Zhang, a new postdoc in my lab. Together, they looked at an additional 3 AD cases by Cryo-EM and another 15 cases by negative stain-EM. They observed the same PHF and SF structures in all cases examined, suggesting that the structures observed are common for Alzheimer's disease. The extent of the ordered core of the Alzheimer fold explains why a mixture of 3 and 4 repeat isoforms of tau are incorporated in the filaments. The ordered core starts at valine 306, which is right at the start of the 3rd microtubule binding repeat. Whether the 2nd repeat is spliced out or not in 3 or 4 repeat tau isoforms doesn't matter, because it falls within the disordered fuzzy code. So whether molecule with or without the 2nd repeat are incorporated into the filament doesn't matter, and incorporation can proceed randomly. How about the other tauopathies that Michel mentioned? What happens if the tau isoform composition in the different tauopathies is different? For example, in Pick's disease, only 3 repeat tau isoforms are incorporated in the filaments. When Ben looked at tau filaments isolated from the brain of a Pick patient, he again observed 2 types of filaments. He called them the Pick narrow filament and the Pick wide filament. The resolution of the Pick wide filament was not high enough to allow de novo atomic modeling. However, you can see from an XY cross-section that it is made up of 2 copies of the Pick narrow filament, which are packed in a head-to-head fashion. The resolution of the Pick narrow filament structure to 3.2 angstroms was high enough, to allow de novo atomic modeling, and revealed this distinctly different shape of the protofilament, now J-shaped as opposed to the C-shaped structure of the Alzheimer fold. In the Pick structure, the 2nd half of the 1st microtubule binding repeat is part of the ordered core. The difference in residues with the 2nd microtubule binding repeat explain why 4 repeat tau cannot be incorporated into the Pick structure, thereby explaining why only 3 repeat tau assembles in Pick filaments. Wenjun then looked at 3 cases of corticobasal degeneration by Cryo-EM too. She also observed 2 different types of filaments, and different cases with the same disease all shared these filament structures. The type 1 CBD filaments are made of a single protofilament. Type 2 filaments are doublets of where 2 protofilaments of type 1 are packed against each other in a symmetrical manner. Again, resolution was sufficient to allow de novo atomic modeling, and this time Wenjun could observe that the entirety of the 2nd microtubule binding repeat, shown in light blue, is part of the structured core of the CBD filaments. And the overall structure of the core, again, is very different from the C-shaped and the J-shaped filament folds for Alzheimer's disease and Pick's disease. Wenjun also observed an additional density in a large hydrophilic cavity, which was unconnected to the density for tau. It appears to be coordinated by 3 lysines sticking into this cavity. This density suggests that there might be anionic molecules involved in the formation of CBD filaments in CBD disease. So how about the tauopathies which share isoform composition? Michel mentioned that we also looked at chronic traumatic encephalopathy, which shares with Alzheimer's disease that all 6 isoforms of tau are incorporated equally. CTE is the only tauopathy with a clear external cause, brain trauma. When Ben looked at 3 cases of CTE, 1 ex-professional American football player and 2 ex-professional boxers, he again observed 2 types of tau filaments. And these again were different from any of the tau filaments we'd seen before. Again, cases with the same disease all shared the same structures of their tau filaments. Compared to the Alzheimer's disease fold, we observed that the same residues, more or less the same residues, form the ordered core of CTE filaments as they do in AD filaments. This explains why also in CTE you get an incorporation of both 3 and 4 repeat tau isoforms. However, the CTE fold is different from the Alzheimer's fold. It is more opened up, and in particular, the tip of the C in Alzheimer's disease opens up in CTE, and in CTE filaments, Ben observed again an additional density inside a now more or less hydrophobic cavity, suggesting that hydrophobic molecules may play a role in the formation of CTE filaments. Together, these structures have painted a surprising picture that each tauopathy is defined by a distinct fold for the tau filaments. In future work, we would like to understand why that is. What drives the formation of this remarkable structural specificity within the different diseases, and what is the role of these structures? In order to be able to study this in the lab, we will need to let go of only looking at brain-derived tau filaments. We need a system where we can apply experimental perturbation and see how the system reacts. So what we really need is an in vitro model system for tau aggregation. A system widely used to date is the addition of heparin to overexpressed recombinant tau. However, when Wendran in my lab looked at both 4 repeat and 3 repeat recombinant tau filaments made after the addition of heparin, she identified at least 5 different filament types. And she managed to solve the atomic structure of 4 of these. These are shown on the right-hand side here. And as you can see, they're generally smaller than the structures observed in the diseases. And none of the structures made through the addition of heparin actually faithfully replicates the structures we observe in disease, indicating an important lack of the ability to make disease-related structures in the lab nowadays. When we compared all these different structures, we observed that the same residues tend to form beta strands, and that the prolines and glycines in which the microtubule binding repeats are relatively rich are involved in providing structural versatility through different conformations of the loop regions in between these beta strands. Together with an observation that we observe a lot of additional densities which do not seem to be connected to tau. And I mentioned the internal structures in CBD and in CTE, but we see similar structures also on the outsides of AD filaments or PIC filaments. And if you compare different cases of the same disease, these additional densities are always there. So based on these observations, we have a current working hypothesis that it is a combination of tau isoform composition with the presence of external cofactors, possibly on the outside of the tau filaments or incorporated like in CTE or CBD filaments, and possibly also the presence or absence of specific post-translational modifications on the tau molecules themselves, which can drive the formation of very different amyloid filament structures from the same amino acid sequence of this tau protein. Now, Michel also showed this slide about alpha-synucleinopathies. Again, there is multiple neurodegenerative diseases caused by amyloid formation of the same protein, in this case, alpha-synuclein. Manuel Schweighauser and Yang Shi in Michel's group isolated alpha-synuclein filaments from 5 cases of MSA. Again, they observed 2 different types of filaments, which they termed MSA type 1 and MSA type 2 filaments. Unlike tau, there are two different protofilament structures that come together to form doublets of filaments in alpha-synuclein MSA. And again, a bit like in tau, there is unidentified density, in this case at the interface between the two protofilaments, that seems to be involved in holding these together. Also, like in tau, the structures that we determined from MSA brain were very different to the existing structures done by NMR spectroscopy or Cryo-EM in other labs from in vitro aggregated recombinant alpha-synuclein. Manuel and Jan also looked at 3 cases of dementia with Lewy bodies and analyzed by Cryo-EM the alpha-synuclein filaments extracted from these cases. These filaments did not appear to twist in the micrographs, which precluded their 3D structure determination. However, the appearance of the 2D class averages shown here is incompatible with the 2D class averages of the MSA filaments, suggesting that what's true for tauopathies may also be true for alpha-synucleinopathies, that different structures characterize the different diseases. That brings me to my conclusions. I've shown you how Michel and I, over the years, have solved structures of tau filaments for 4 different tauopathies and now also 1 alpha-synucleinopathies. How each disease forms different amyloid, how different amyloid structures are formed in each disease, and how our currently available in vitro aggregation methods do not replicate these structures yet. This has taught us a surprising finding that unlike globular proteins, which may have evolved into a very specific shape to fulfill a specific function, amyloid structures appear very structurally versatile. The very same protein sequence can adopt many different structures. It remains to be seen how much this will hold for other proteins that are known to form amyloid structures, and what the relevance of previously determined in vitro aggregated structures of these proteins are. For me, having started out as a developer of algorithms for the processing of Cryo-EM images, it remains extremely exciting that we can now do atomic resolution structures of amyloids that are extracted from the brains of patients. And these methods will be applicable to many more diseases. I've mentioned most of the people on this slide. Jasenko was involved in algorithm development to increase the resolution of the CTE filament structures. Dino, Masato, Ruben, and Kathy are our collaborators who provide brains, filament samples, and more. Tony, Alexei, and Gareb have provided invaluable advice throughout all these projects. And all of this work is in very close collaboration with Michel Goedert. And this is why we decided to do this seminar together today. Thank you very much for your attention.
Thanks so much, Michel and Sjors. That was a fantastic presentation. We have a few questions here, and I want to start with, you alluded to, Sjors, the fact that there were advances in Cryo-EM that facilitated being able to decipher these atomic structures. I'm sure this is a whole separate talk, but can you sort of briefly tell us what those advances were that allowed this?
Yes, importantly, this was the availability of direct electron detectors, which increased signal-to-noise ratio in the raw Cryo-EM images significantly. And Richard Henderson, who will give the culture talk, was actually involved in the development of these. So that's one part of that. Another part is also the development of more powerful software. And for example, the Bayesian approach in the Relance software is one example of that.
Great, thank you. I think we're gonna go to some audience questions. Excuse me. Bin Zhu has a question for Michel. Bin, are you there?
Yes, I'm here.
Yes, I'm here.
Go ahead, Bin.
I have 2 questions for Dr. Goedert. First, since there are 6 tau isoforms, is it possible to quantify each of them, for say by mass spec in the different subregion of postmodern AD patient brains? Because the reason is these 6 isoforms are highly homologous. So just wondering whether it's possible, the challenge are. The second question is, do you believe that those repeating regions like 3R or 4R, they are the main contributor to the neurotoxicity of the tau, not the fuzzy regions? And related, is the straight filament less toxic than PHF filament?
Okay, thank you very much. Yeah, you could in principle, I think, by mass spectrometry, distinguish between the 6 isoforms. You could also, in principle, make antibodies that distinguish between them. At the moment, what is known is that there are 4 major tau isoforms in the brain and 2 minor ones. And the minor ones are those that have 2 N-terminal inserts rather than 1. Whether at individual cells you could see differences in these different types of isoforms is not really known. So even if you looked in subregions of the brain, there would still be millions of nerve cells in each probably. So at the moment, it's not clear whether different expressions of different isoforms in particular cells could contribute to the assembly of the protein into filaments. For instance, it's unclear why there should be tau diseases where you have 3-repeat tau in the filaments, like in Pick's disease, or 4-repeat tau, like in corticobasal degeneration, if all cells express all 6 tau isoforms. So that's not known. As to your second question, I would think that probably it is the assembly of tau into abnormal structures that causes the toxicity, because there is quite a good correlation between the presence of tau inclusions, which are made of these abnormal filaments, and neuronal cell death, certainly in Alzheimer's disease. So I would have thought that it's probably the repeat region, which is the core of the filament, that causes the toxicity. As to the relative toxicities of straight and paired helical filaments, I mean, I think that's an important question, but nobody knows. At the moment, there is no way to distinguish between these 2 types of filaments in any histological or other way, only using cryo-EM, basically.
Thanks, Michel. We have a question from Eric Zhao. Eric, are you there?
Yeah.
Go ahead, Eric.
Can you hear me? Yeah.
Yes.
Okay, especially for the inheritable tau-mediated disease, there may be point mutations of tau that trigger its aggregation and the formation of tau filament. So do your resolved structures of the patient-derived tau filaments provide some insights into the mechanism of the mutation-triggered filament formation or aggregation of tau protein?
Shall I start on this one?
Yes, please do, Michel.
Okay, so I don't know. I mean, we are working at the moment on trying to determine the structures of tau filaments from diseases where single amino acids are mutated in the repeat region that cause familial frontotemporal dementias. We don't know yet. As far as I can see, there is no obvious way to understand the effects of these mutations using the structures made of wild-type tau assemblies that we have determined so far.
We have another question from Ellie Johnson. Ellie, are you there?
Yes, I'm here.
Go ahead.
Hi. Really interesting talk, especially about the densities. I'm just wondering whether you think the cofactors are mediating the interaction.
Ellie, we're having a little problem hearing you. Could you try to speak louder? And if not, I will go ahead and read your question.
Okay, can you hear me now?
Not really.
Okay, go ahead and read it for me.
Okay. Ellie is asking, do you think that the cofactors are mediating the interaction surface of alpha-synucleins that you showed with the extra density, and are other cofactors necessary for filament formation?
Yes, we do not know. We don't even know whether these are necessary for filament formation, or all that we observe is structures that have accumulated over decades, and we see the end result of disease after the patient dies. What we would really like to know is how the process started, and what factors play a role. But for this, I think a lab-based system will be very important to develop.
A quick follow-up question on that I think a few people have is, do you have any ideas what this mystery density could be? And do you think it could possibly be the same molecule in each of the filaments?
We think, so we see densities at the interface of straight filaments of tau, AD, we see them inside CBD filaments, and we see them inside CTE filaments. So the CTE density is probably not the same as the CBD and the one at the interface of the straight filaments in AD, because the CTE environment of the tau molecule that the tau molecule provides is mainly hydrophobic. Whereas in both CBD and at the interface of the AD straight filaments, these are lysines coordinating. So it could be that the one in AD and CBD is a similar molecule, but probably not the one in CTE.
Great, thank you. I'm going to, let's see, I'm going to ask one last question that I see here, and it's sort of like a broader question that I'm sure many people are wondering. Now that we know the structures of these proteins in Alzheimer's and other diseases, how possible is it for us to create some sort of inhibitor? Do you think that's on the horizon? Do you think that will be particularly, challenging, or do you think that's something that will happen fairly soon?
Perhaps both Michel and I can give our view on that. I think, again, having a working in vitro system that we know replicates these structures will be important if you were to design some molecule to inhibit filament growth. I spoke once to a pharmaceutical company who used heparin to grow tau filaments, and then did many compound screens to find molecules that would inhibit tau filament formation. That probably is not the right way to go. Perhaps Michel could add.
Yes, I agree. I think in order to understand the filament formation, one needs to know what these possible cofactors are. And in order to develop inhibitors that are relevant for the human brain, you need to understand what happens in the human brain. So you need an in vitro system to produce filament morphologies and structures that are the same or very, very similar to those in the human brain.
All right, thank you so much, Michel and Sjors. That was fantastic.